Good morning, students. Yesterday we have done a reteach of the Punnett squares, and if you still do not understand the concept, do not worry, we still have plenty of practice to do. Today we're going to learn about genetic disorders. And so right now, most important idea is that sexual reproduction allows for possible combinations of genes. That is what a Punnett square is to show you. Two parents combine half of their genes, half of their traits, in order to create an offspring. That is a possible combination. Genes are segments of DNA, and so we're passing traits from one generation to another, from parent to offspring. And behavior objective, once again, students are to show integrity and academic honesty. Moving on. Now, some of you might realize that this is question 2 from yesterday's assessment. Many of you do not seem to realize what a heterozygous cross means. For example, in question 2 for yesterday's assignment and the assignment from 2 days ago, heterozygous literally means a hybrid. So when you write the genotype of a hybrid, it should be a mix of a capital and a lowercase letter. That is as simple as it is. And so, this is a Punnett square of two parents that are heterozygous. Both of them are heterozygous. This is a chart showing the genotype of the parent, percentages of the child's genotype, and the phenotype. Now, some of you do not seem to understand what dominant and recessive means, the role that it plays in phenotype. Phenotype means the expression of the code, of the genotype. So in this situation, homozygous dominant, heterozygous, if there is one dominant allele, the dominant trait is expressed. Recessive traits are expressed when there is no dominant. And so in this example, there are no capital B's. So that means black, which is dominant, is not there. You don't have the trait. You can't express black fur. You have two alleles for brown. Brown is recessive. So there you go. You have brown fur. And that is the review for the actual question 2 of yesterday's assessment. And so now we're going to go back to the big question. How do genetic traits get passed from generation to generation? And so in order to pass on DNA from one generation to the other, your body makes cells. It can make body cells, or it can make sex cells. Sex cells are used to create the actual offspring. So sex cells, when combined, create the next child. Body cells are just regular cells in our bodies. They make copies of each other. And so in reproduction, DNA is copied over and over again because you need to make copies. As you make more cells, each cell needs a copy of that DNA. Body cells get one full set of DNA. Sex cells, sperm and egg, only gets half. And so, just like when you use a photocopier, a copy machine, errors will appear. So right here in this example, this line is not a straight line, so there's a slight little error that was made. And so in there, just like printing, errors can arise. Same thing in DNA. As you make more and more copies of DNA, errors will happen, changes will happen. But I don't want you to think all errors are bad. So not all errors are bad. But then first, we're going to talk about errors that really does mess up the genetic template of the individual. And so when there's a really bad mutation error, it can lead to genetic disorders. And so I have 3 examples right here. First one is cystic fibrosis. Cystic fibrosis is a disorder where your body makes incredibly thick mucus. So right here in this picture, right here, this is inside your lungs. There are tunnels. There are passageways where air flows through in order to make sure that gas exchange can happen. But when there is mucus not being carried away, removed, these passages get clogged up. And so people with this disorder have a harder time to breathe, and it can lead to respiratory lung infections. Now, the next genetic disorder is sickle cell anemia. Now, red blood cells, if you look carefully right here, are usually circular. But when you have sickle cell disorder, your cells look more like a crescent moon. It's like a sickle. It is like a C shape. And now, the reason that circular blood cells are great, because circular blood cells can just fit right through the blood vessel. No issues. But it's like bumper cars. If you bump into someone, you just keep going. You just keep moving. But for sickle cells, because of the shape, it can get stuck in areas of your blood vessel. And so this can lead to blockage, which can actually block blood from reaching an area, or it actually can reduce the amount of blood that can reach an area. So there's not as much blood going through this vessel. And so that can lead to a condition called anemia, which means shortage of blood. Now, these 2 examples, cystic fibrosis and sickle cell anemia, they are autosomal recessive disorders. And so you all know what a recessive trait means. So these disorders are recessive. So in order to have a disorder, both of your alleles need to be recessive. But if you're a carrier, which means you only have one allele that is recessive, you don't have the disorder, but you carry an allele for the disorder. So you actually can be a carrier and not have the disorder, but you run the chance of passing this one allele to your children. Now, there are some dominant disorders. A very famous one is known as Huntington's. Huntington's is a nervous system. It's a degeneration disorder where you're born with a perfectly fine nervous system, but at around 30 to 40 years old, you actually start having your nerves start dying. And so this results in this picture right here, you lose control of your own mobility, your coordination, and even your mental faculties. And so you can have slurred speech. You can have not so lucid moments. And so a dominant disorder means having just one allele. It is rare to have both. It is actually really rare. Having one copy of this error will result in a disorder. And unfortunately, this disorder is fatal. all you can do with medicine is to have improve the quality of life the person with this disorder has. There is no cure for Huntington's. Now, this vocabulary slide has all the vocabulary that I have covered. But the most important one I want you to understand is the word mutation. It is, most people think of errors, but it is actually, a mutation is a change to the genetic material of an organism, and it can be good or bad. For example, sickle cell anemia, actually, if you have this disorder, yes, it's bad, but if you're a carrier, you actually have a better chance of surviving malaria. And so a mutation is good or bad. And so my takeaway message is mutation. It is a change to the DNA. Just a random change. Now, an important element of sexual reproduction is you cannot control which chromosome an organism inherits. We have not reached the point in science where we can say you will inherit this. No, there is a whole ethical issue in here because then humans are playing God. So I'm just going to step away from there. So this is why a Punnett square is important. It's because there is probability. It's random. There's a randomness to reproduction. And so we're going to the example here for sickle cell anemia. If both parents heterozygous, both parents are heterozygous, so they carry one recessive and one dominant. So they have one perfectly fine copy, so one good one, capital S, dominant, that's good. Recessive, that is the issue, that is the error, that is the mutation. Now, two heterozygous parents have an offspring. This is the possibility that the child can have the disorder. This is a autosomal recessive disorder. That means the offspring must be homozygous recessive in order to get the disorder. And so, if you do a Punnett square of two heterozygous parents, you have 25% chance of homozygous dominant. That means perfectly fine. You have a 50% chance of a heterozygous offspring. Now that's a carrier. Perfectly fine, but that offspring has one mutation, mutated allele. But he or she is still fine. Now this is the issue. 25% chance the offspring is homozygous recessive. That means you have the disorder. 25% chance if both parents are heterozygous. Now, there are other situations. What if someone with the disorder have a child with a perfectly fine individual, a homozygous dominant? Now, those are possible questions we can ask you. And so, my takeaway message is this table is to show you that when two parents have an offspring, depending on what is their genetic history, what is their genotype for certain disorders, you can determine the probability of their offspring having the disorder. And so in this case, two parents who are heterozygous, they have a 25% chance of having a child with the disorder. And so for today, there will be two videos that will explain more on genetic disorders and how it gets passed on. It is just going to reiterate what I say and go more in depth. And so after you watch these two videos, there will be an assignment for you to complete. It is another Punnett square practice. And so yesterday, I have shown you a step-by-step approach to the Punnett square. Today's assignment, you will read the question carefully. I cannot stress this enough. The first step to success is to figure out the genotype of the parents. You have to read the question carefully. And so, good luck. Also, first page of assignment is just a review of what we have covered.